to introduce Dr. Bettie Sue Masters. She's a Robert Welch Distinguished Professor in Chemistry at the Department of Biochemistry at the University of Texas Health Science Center at San Antonio, Texas. The title of her talk will be Molecular and Cellular Effects of Human Polymorphic Variants of NADPH-Cytochrome P450 Reductase. Dr. Bettie Sue Masters.
All right, so it's a long title, and this is a story sort of my career. And I'm not going to talk about nitric oxide synthase today because actually this is a revisitation of my early life. So it'll be fun for me to tell you a story that's just now evolving and for which we have just received new funding from the National Institutes of Health. So it's one of those things that what goes around, what comes around, goes around, or goes around, comes around, whatever it is. Here is my group. We often forget to recognize the people who do the work in our labs, and I'm going to be mentioning many of these people's names during my talk. This is a student now, then a postdoc, and now a postdoc at Vanderbilt University who contributed to the work I'm going to talk about today. And where is, I guess, Chris made this slide, so he didn't put his own picture up here, but I'll show you his picture later. Yuzuru Ishimura was the president of the Japanese Biochemical Society and the chair of biochemistry at Keio University in Japan, and decided when he retired that he wanted to come and do a postdoc with me. So he spent 3 years in my lab, and it was wonderful because we had actually been affiliated with one another at Southwestern Medical School in Dallas many, many years ago, before some of you were born. Tom Shea runs the lab. Keeps everything running. This is a student who actually has taken an interest in research, and I hope we can capture him into the research arena. Mark Panda and Ying Gao are both postdocs. Mark is now a faculty member at University of Texas at San Antonio. Linda Roman has done a yeoman's job on the nitric oxide synthase problem. I will not be talking about her work today. Debbie Earle has a halo on her because she passed away unexpectedly last year. She was a dear friend and confidant and was my administrative assistant. So the story began. Do you recognize who that is? This is the starry-eyed graduate student in University of Sheffield in England in 1962. And I am holding in my hand a test tube which has NADPH-Cytochrome P450 Reductase in it. And I had worked for 6 months constantly preparing this enzyme from pig liver, because pig livers are big, right? So we had to make a lot of this stuff to go over and visit with Vince Massey and Quentin Gibson, Graham Palmer, Gregorio Weber, and some of the most famous biochemists. You must have recognized these names, to learn how to do spectroscopy, to learn how to do stop flow spectrometry, spectrophotometric measurements, and even some EPR. So this was the beginning of my career, and I stuck with it. So I'm bringing this slide because I know there's some young women in the audience, and I just wanted to, this was an inspiration for me, and I wanted it to be an inspiration for you. There was a book written by Eve Curie, who just passed away actually this past year at 102, I think, about her mother, Marie Curie. And the quote goes as follows: "The idea of choosing between family life and the scientific career did not even cross Marie's mind. She was resolved to face love, maternity, and science, all three, and to cheat none of them. By passion and will, she was to succeed." That has been certainly my goal. I'll never be Marie Curie, but she certainly had some wonderful things to say about her mother. Now, what I'm going to be talking about is monooxygenase reactions, and actually an enzyme that contributes to them very similarly. And monooxygenase reactions are those which incorporate one atom of oxygen into an organic molecule. This was a heretical concept until the mid-1950s. In fact, there were great arguments in the literature, people calling each other names because that couldn't happen. Actually, it was Howard Mason's laboratory and Osamu Haishi's laboratory, which proved that this did happen in bacterial and mammalian species. So what you see is the oxygen atom actually being split, one atom going into water, the other one being incorporated into the organic molecule. But what I'm going to be talking about is this enzyme, NADPH-Cytochrome P450 Reductase, a boring old redox enzyme that has two flavins on it, one FAD and one FMN, which we discovered in our laboratory at Duke University when I was a student. And the electrons that go into the cytochromes P450, and last count, there were over 5,000 cytochromes P450 identified in all of phylogeny. We don't have any idea what all of those do, but we know that they're involved in a huge variety of reactions, all of which involve monooxygenation. Now, there are 2 electrons required for this process. The first one goes in so that the cytochrome P450 heme, which is a separate protein, of course, is reduced so that oxygen can be bound. The second electron is required to actually dissociate, cleave the oxygen molecule. So when I was a student, I worked very hard on the mechanism of NADPH-Cytochrome P450 reductase, but we didn't call it that because we didn't even know it was cytochrome P450 reductase. We called it NADPH-Cytochrome C reductase. Well, there's no cytochrome C in the endoplasmic reticulum, so we didn't know what its function was. And so there were hints in the literature that perhaps this enzyme should be doing something else, since it wasn't in the mitochondrion and it was in the endoplasmic reticulum. So we decided, when I moved to Southwestern Medical School, I had a very generous mentor who gave me this problem. I walked out of the door with pure protein and was able to do some experiments the minute I walked into the door at Southwestern. And so we made some antibodies. And we decided, okay, let's throw these into some gamiches and see what happens if these are inhibitory antibodies, see what happens. So we did, and the labs of Omura in Japan and John Shinkman, now at Connecticut, was at Yale when he did these experiments, threw in antibodies and showed that there was a concomitant inhibition by these antibodies of in this case, this experiment was done with aminopyrene demethylation, cytochrome P450 reduction is measured spectroscopically, and cytochrome C reduction. So there was a concomitant inhibition. That meant that this enzyme was involved, at least in this case, in drug metabolism. We also did comparable experiments, I'm not going to show you all that history, with steroids and adrenal microsomes. So this slide is just simply to point out, because this will become pertinent later, that there are a number of P450s involved in steroid hormone metabolism. In fact, it's absolutely required. The women and men in this audience are who we are because of steroids. And so these actual differentiations occur because of these enzyme activities, the 17-hydroxylase, the 1720 lyase, and the aromatase, which is, of course, the enzyme responsible for the aromatization of estrogens. So the other thing that's important to know before I get into some science here is what these P450s do in humans. Now these are the human endoplasmic reticulum enzymes that are responsible for a variety of xenobiotic, we call them, xenobiotic metabolizing activities. And you can see here that if you add up the CYP3A4, the CYP2D6, CYP2C9, and CYP2C19, and 1A2, you get over 95% of human drug metabolism is carried out by these few CYPs, cytochromes P450. So they're important. So in the intervening years, I've now spanned 30-some, 40 years, the properties and mechanism of NADPH-Cytochrome P450 oxidoreductase, which I'm going to be calling CYPOR probably throughout the talk, continue to be studied by our lab as well as a number of others, notably that of Charles Casper at the University of Wisconsin, who's now retired. In 1997, our work in collaboration with Jungja Kim, whom Art Haas has mentioned as a collaborator of his. She's an excellent X-ray crystallographer at the Medical College of Wisconsin. She and I then solved the structure of CYPOR and revealed for the first time the spatial contiguity and apposition of FAD and FMN in that flavoprotein molecule. And this is what, of course, we all think our structures are beautiful, but I really think this one's beautiful. You can note right away the very clear domain structure of this protein. The fact that there is a domain that binds FMN, there is a domain that binds FAD, and also NADPH, because the electrons originate from NADPH into this FAD and then flow into the FMN. And that there is a connecting domain that allows some flexibility between these domains. The actual space between, distance between these two isoalloxazine rings, allowing that electron transfer, is about 3 angstroms. Now, what really brings me to talking to you today is the discovery by first Casper's laboratory, whom I just mentioned, the beautiful work of Anna Shen, showing that multiple developmental defects occur and embryonic lethality occurs when you knock out NADPH-Cytochrome P450 Reductase. So that tells us right away it's a pretty important enzyme. But there's some other things that are interesting about this. If you do a conditional knockout, there was other work done by a group in the UK led by Roland Wolff, in which there was identification of novel roles of the cytochrome P450 system in early embryogenesis, effects on vasculogenesis and retinoic acid homeostasis. So I'm going to tell you some more things, but one of the interesting, or several of the interesting comments that I can make about these mutants is that the embryonic lethality was, of course, remarkable at embryonic day 9.5, but there were skeletal malformations in these animals. And also, they had lost, as I just said in this paper, the CYP26A1, which is a vitamin A metabolizing enzyme. So here is the little electrical system here that allows the transfer of electrons, again, from NADPH through FAD to the FMN to the P450. And here's the structure shown below. Now here is the picture, unfortunate picture, of the Antley-Bixler Syndrome. This syndrome was discovered in 1970, or reported in 1975, and shows a trapezoidocephaly, midface hypoplasia, and cartilage abnormalities with multiple synostoses in the cranial region and skeletal fractures. These children can survive for a certain length of time if they undergo surgery, but because of the synostoses, the brain cannot develop, so they have to have brain surgery to survive, and often do not survive past puberty. So this gentleman is Walter Miller. He's a pediatric endocrinologist at the University of California in San Francisco. And we happen to, and I say we, I have to give credit to Dr. Pavel Marticek, who is a Czech clinician scientist who comes to my lab about 6 months of the year, off and on, back and forth, and has done so for the last 13 years. He's worked on our nitric oxide synthase project, which I've been very much involved in since I went to San Antonio. But Pavel reads the clinical literature. And actually this paper was published, and he saw this paper and called it to our attention. So we all hopped on it and read it, of course, and it became very exciting to us because it looked like this enzyme was causing disordered steroidogenesis. And it had some, depending on the mutations, it had some effect or no effect on looking like Antley-Bixler Syndrome. Now, the number of polymorphisms that have resulted or have been discovered by Dr. Miller's group are phenomenal, and I'm going to show you that in a moment. But let me just show you some of the data that he produced. Remember, I just showed you this electron transport chain, right? Starting with NADPH and going through. He has shows 4 individuals on this slide showing mutations, specific site-directed mutants. We, all in this room, there's probably a huge representation of these polymorphisms in this room, many of which don't affect us at all. But some of these are, and all of these actually on this slide, have some serious consequences. And so we looked at these and we looked at the structure. You know, we're biochemists, right? So we took a look at the structure and we said, you know, no wonder these must have some functional reason. And so here, this is scary, but here are the polymorphisms that Dr. Miller has actually identified in human patients. They have done 800 patients now at UCSF in a big program that they have gotten funded from NIH for. And all of these have popped up. So we are in the process, and the funding that we have just received, of characterizing a number of these polymorphisms, and we have a plan, and we'll get to that later. The 2 that I have boxed up here, we have already published, and I will show you some of that data in a moment. And we're now working on this one, and we have 5 more actually bubbling up in the lab as I speak. So what we did was we bacterially expressed the human CYPOR, and we did it in several forms. As we all know, membrane-bound enzymes are really difficult to deal with. Fortunately for us, the NADPH-Cytochrome P450 Reductase has a hydrophobic tail, which the only function for that hydrophobic end is for it to attach to the ER. There are no other hydrophobic segments in this molecule that are needed for it to attach to the ER. So we can make the soluble form of the enzyme, and that is actually the structure that I showed you in the previous slide. This is the holoenzyme containing that. So we're making both of these. The problem with the soluble enzyme is because it cannot attach to a membrane matrix, it will not interact with P450s. P450s are also hydrophobic. So we can do a lot of things with the soluble enzyme, but we can't reconstitute it. So that's why we make both forms, and we can do a lot of characterization enzymologically of the soluble enzyme. So here are the 2 mutants here that I'm going to emphasize today in my talk, and one of these is the V492, which is comparable to the V489 in the rat protein, and Y459. You can see with the FAD molecule shown here, focused in on, the isoaloxazine ring, you can hardly see it here, but to make this clear, we oriented it this way. This is a 3-ring structure that's going back into the blackboard. This is obviously a stacking residue for, and a pi-pi interaction occurs between this isoaloxazine ring and this tyrosine residue. And this residue actually hydrogen bonds with the FAD. So we began to examine these. So we prepared them, and as you can see, the wild-type enzyme has a different color. These colors are not true. This is not what your eye would see if you were looking at the tube, but I just want to illustrate that the colors are different. And if you look at the spectrum, you can see, yeah, these are really different. The 492E has much, much higher semiquinone in this region of the spectrum. This is the wild-type spectrum, as you would see in semiquinone form. So what we did then was, all right, if there is a residue that we've mutated and we want to see what it does and it's made a difference in the spectrum, could it be that there's a perturbation of the flavin binding? And sure enough, if you examine the binding here, this is with micro-BCA showing more protein than flavin with the 459H and the 492E, and with the wild type equal concentrations. Over here with FPLC, you can see with FAD separation, FAMN, wild type having both, 459H and 492E, very little FAD. So in looking at the functional assays, you can see here that the wild type with the soluble form has a very nice catalytic efficiency, but the 492E tenfold less. The wild type holo enzyme, very good catalytic efficiency. The holo is the 459H and the 492E much lower. So then when we examined the activities toward a reconstitution system, and remember I told you we can only do that with the holo enzyme. So we reconstituted prostaglandin E1 hydroxylation with these enzymes to see how they could reconstitute. You can see here that with the wild type enzyme one gets PGE1 omega hydroxylation, but not with the 459H or the V492E. However, we decided, let's see if we can add FAD back. We didn't have any idea about affinity, so we added back, here's the FAD and FMN separation, wild type and so on. Here's the addition of FAD to each of these, and you can see in this slide that the FAD has been added back, and we can get hydroxylation of PGE1 in both cases. So we can rescue these two proteins by adding back FAD. So these are severe mutations, these two. The patients do suffer from Antley-Bixler Syndrome, and so one can ask the question, and I'm planting this in your mind now, what can be done about this, perhaps prenatally, if you could predict it? So keep that in mind for a moment. This is Jungja Kim, and she has now, the structure that I showed you earlier was the rat enzyme. We now have structures of all of these human proteins and mutants. She's writing the paper, I'm beating on her now, to write the paper, and Art's smiling because we know that Jungja does things in her own time. But that she will be the first to have the human enzyme, and also she will have all of these mutant structures, and we're very excited about these. Examining much more closely what is happening with the V492E, just as an example. Remember, that was the hydrogen bonding one. That wasn't the pi-pi interacting residue. You can see that there's a disorder in the location of that residue. We don't really know why that's so severe, but it is a dislocation. And so this apparently is enough to affect the FAD binding in that protein. And here's a much sort of stepping back picture. What was interesting to us is that Jungja called us up and said, there's nothing wrong with this protein. And we're saying, huh? You know, well, it doesn't work and it doesn't bind FAD. What do you mean there's nothing wrong with this protein? She said, it looks just like the wild type. Well, closer examination showed that there were two molecules in the cell. And so what you see is that molecule A is in one configuration with E492, and in the other molecule is in another entirely different configuration. So it is something related to that positioning in this map, and you can also see that the densities, the electron densities, are quite different. This is the one that looks like wild type, this is the one that doesn't, And you can see that the density is very different along that green ribbon. All right, so we are collaborating with people who know how to do things that we don't know how to do. And I don't know how many of you know anything about protein film voltammetry, but it's a new way of looking at redox potentials. And folks like us who deal with redox enzymes have to know how these electrons are flowing, and of course that's part and parcel of the whole thing of how they work. Dr. Sean Elliott actually was trained in England on his postdoc, is now at Boston University as an independent investigator, and this technique, this protein film voltammetry, allows you to coat electrodes with the protein and measure kinetics. Now this slide does not show you the kinetics because we haven't done them yet, but that's our next step. Here we've been able to show that we can actually measure the redox potentials of these proteins on those electrodes directly. They can be gold or graphite or any number of things, and not lose the prosthetic groups. And that's a very important issue. So Sean is helping us do that. He's part of the grant that we just got, and we're excited about the future on that. All right, here's Chris Moronic. Chris deserves a lot of credit. He's a postdoc in my lab right now. He's on his way to becoming a young first faculty member, and he's going to go in for one of these R00 grants because he's qualified for it. He's shown here with Michel Kranendonk, who is from Portugal, and he's another collaborator. What these slides show, the reason that Chris deserves credit is he was very seminal in the writing of the grant and thinking through this process. He's very good. So at the top, we are using a bacterial mutagenesis assay and a bacterial system to express these things in to mimic the endoplasmic reticulum in mammalian systems. There is a ratio of the flavoprotein to the CYPs, the cytochromes P450, in these systems of 1 to 5 to 20. In other words, there are lots of P450s compared to the number of flavoproteins. And in a reconstitution system, all of the ones that have been done up to now have been done on a 1-to-1 ratio. So that doesn't show you what's going on in the endoplasmic reticulum. Dr. Kranendonk has designed a bacterial strain in which he has been able to express these proteins in the proper ratio so that we can actually measure these the way they would be in a mammalian system, and also to put in not just one P450 but a variety of P450s and let them compete with one another. So these assays are all set up to do that, and you can see here in the testing of CYP1A2, the ethoxyresorufin O dealkylation is affected. This is the first time that it's been shown that drug metabolism is affected by these mutants. Well, okay, you would expect that to be the case if you have a mutation that is crippling the transfer of electrons from the flavoprotein into the P450. There are other subtle polymorphisms that won't be that straightforward. It won't just be an electron transfer thing. It may be a protein-protein interaction mutation. So this is where we're going. The bottom of this slide shows a mutagenicity assay. I know many of you have heard of the Ames test. It was done to test every compound in the world, including hairspray. All right. Well, this is sort of an Ames test. And you can see here that these mutants were not able to mutagenize, whereas the wild type, very, very capable of upholding or supporting mutagenicity, these mutants were not. And interestingly enough, there was a different, I'm not showing this on this slide, but there's a differential effect of FAD readdition. In one mutagenesis assay, not in this one, but in one mutagenesis assay, it was able to regenerate it, rescue it, but in the other it was not. So that's kind of an interesting phenomenon. We don't understand it, but that's the case. Now, the next thing that we need to do is to understand why it is, going back to the original observations, why are these skeletal defects occurring? What does that have to do with cytochromes P450? Is it just related to retinoic acid metabolism? Is it some other signaling phenomenon that we're examining here. So Dr. Satya Panda, who, as I said, was a student in the lab, is now at Vanderbilt, did the original experiments in which he knocked down CYPOR in a human osteosarcoma cell line, showed that he could knock it down, and then looked with the microarray technique at some candidate genes. So that's where we are at this point in time in examining these, and we're also looking at other cell lines so that we can determine what happens when you knock down CYPOR in a developing situation. The other thing that we've got is we have a third, fourth collaborator who actually generates knockout mice and has some CPR lox mice. And we intend to carry that forward and look at what happens when we introduce these mutant CYPORs into these mice and look at the tissues that result. We can do that actually after the fact with using the lox animals, or we can actually introduce the genes into it. And this is a whole new area for me. I mean, I am not not a molecular biologist, nor am I a geneticist, but I find it very exciting. So here's the approach that we're going to be taking. We're looking at the data from the clinic. We have all of Dr. Miller's mutations. He is his full collaboration. He sent us all of his clones, so we have perfect freedom to develop these things. But you can't do them all 40, so we have to use some kind of a scheme to decide how to do this. So we'll look at severe to mild, make choices of those particular polymorphisms that we want to look at. We will do the molecular and biophysical characterizations, but we also want to get into the cell modeling. Finally, here's the group again, and here's Chris back here, very important. This was when we were celebrating Satya's getting his PhD. And this is my home team, my family. My husband, Bob, who you will see and meet at this meeting, and my daughters, Deborah and Diane. And a very important occasion for me in Prague 2 years ago. And I want to thank NIH for some grant support, and the Czech Republic for Pavel Martasek, per se, and for his grant support.